Hello, my name is Nigel Brooks, and today I'll be talking about my protein and its mutation. The protein that I did a model of is Piezo1 protein. Piezo1 protein is a mechanosensitive ion channel receptor. Its main work is to detect the mechanical change in cellular environment configuration and relay this inside the cell through variations in ion flow across the cell membrane. Basically, it senses and converts mechanical forces into chemical and biological signals influencing cellular responses. The expression of Piezo1 takes place on the plasma membrane of cells, where it mediates in mechanosensitive ion conducting. Cells that are included are endothelial cells lining blood vessels, red blood cells, epithelial cells, and sensory neurons. Highly expressed in red blood cells, these aid the cells in sensing the flow of blood and are important for maintaining the structure of the cells. It is a mechanoreceptor that senses physical forces and transduces them into intracellular signals, usually with calcium ion influx. This classification is highly relevant because Piezo1, like other receptors, act as a kind of gate. It perceives external stimuli, which trigger an intracellular cascade of signals, resulting in cellular behaviors such as migration, proliferation, and differentiation. Now I'll be talking about the mutation, which is R2456H, and it is generally considered an activating mutation in the Piezo1 protein. This mutation changed one arginine to histidine at position 2456 and modified Piezo1's response to mechanical stimuli through a mechanism that includes a reduction in the inactivation rate. Thus, Piezo1 stays open for longer than it typically would in response to a mechanical stress, allowing a prolonged influx of ions, especially calcium. Now I will be talking about my model, which I made out of Play-Doh, and each color represents a different part of the protein. So to start off, the teal color represents the blades in the Piezo1 protein, and the pink little part represents the cap of Piezo1. Then over here I have these three little purple Play-Doh balls, which represents the ions. So when the cap opens, it lets small ions in, and that allows the function to carry out its processes. Also, I only have one model because when Piezo1 is mutated, the structure doesn't change. However, the function does. Now I will be talking about the limitations that I've had with this project and the model. The first one is that it isn't as detailed or to scale as a regular Piezo1 or mutated Piezo1 protein. The second one is it doesn't depict the movement of the protein because it's just Play-Doh. And then that leads to the third one, which is it is made out of Play-Doh, so it isn't as strong or real as the actual protein. So that is all that I have to talk about for Piezo1 protein and its mutation.